Hello, everyone, and welcome to the Natural View. Maggie and Todd here with our guest, Brendan Kesler, R&D Innovation Director for Van Drunen Farms and FutureCeuticals, and also postgraduate researcher at the Rowett Institute. Welcome, Brendan. Always great to see you.
Hey, Brian, thank you so much for having me. It's always good to be here. So, yeah, thank you very much.
Yeah, so you've been our guest before. Over the past few years, you've been keeping us up to date on research that explores the relationship between fiber-rich fruit consumption and health outcomes. And I know there's more to know. So today you're going to get us up to date on some of the latest. But first, let's start with some background just to kind of set the stage. So there's an issue with a gap in dietary fiber consumption, right? Can you kind of talk us through some of the causes and concerns there?
Yeah, definitely. And again, thank you so much for having me. It's always really a pleasure to be with both of you. So, yeah, a really big part of my personal research or kind of academic research is focused heavily on dietary fiber consumption gaps. So, in the United States, this is probably one of the most kind of extreme areas where we see a significant gap between what we know we should consume in terms of dietary fiber and what the U.S. population actually consumes. So the latest data we have on the United States is about a 95% gap across the U.S. population. And if we just kind of peel that onion back maybe one layer deeper and ask kind of why people do not consume enough fiber, it's really directly attributable to the fact that people do not consume enough whole fruit, vegetables, and whole grains. So that really is kind of the main cause of why we have such a fiber gap. You know, it's interesting, when I started my PhD, I decided to do that in the United Kingdom. And so I had the chance to start exploring kind of some global data on this as well. And it's interesting, I think, to the best of my knowledge, for any country in the world that has a recommended amount of dietary fiber that the population should consume, and kind of a national survey that tracks that, this is really a global issue. So it's not just impacting the United States, but really any country that tracks fiber intake shows a very significant deficit. And so in the United States, this has been classified, I think, for the last 10 years as actually a dietary component of public health concern, which just means we have very strong evidence that dietary fiber is very beneficial in disease prevention, and we know the population is not consuming enough. So we're eager for the next round of dietary guidelines to come out and hopefully get an updated snapshot of that data. Hopefully the number is below 95%, but I imagine that will still be a very significant gap for the future.
Yeah, I think you're going to have your work cut out for you, your job safe for a long time.
Agreed.
Something. So I'm wondering, we hear about the fiber gap, right? Especially in our industry, and we know there are supplements out there that try to target that with singular ingredients. Can you talk about that a little bit, that issue, and maybe compare and contrast that with NatureKnit and how that can uniquely help fill that fiber gap?
Absolutely. Yeah. So I think a really big piece of my research interest is in kind of what we can do to fill the fiber gap. And so one of the kind of the key questions I have is to your point, Todd. So we know we have this fiber gap in the United States, this same fiber gap globally. There are, one way that consumers try to fill that gap is by going to a local drugstore or Amazon or, shopping and buying a fiber supplement. And so I personally, I don't think there is any such thing as a bad fiber necessarily. But what I do think, and kind of what my research kind of leads to, is that I do think there's a really big difference between purified, isolated, kind of single source fibers that the food or nutrition industry tends to kind of put forth versus the type of real food fibers we get as part of just a whole food diet. So maybe to say that kind of in a bit of a different way, if you woke up this morning and you had a big bowl of oatmeal with nuts and seeds and fresh fruit, that is a very different kind of type of fiber, diversity of fiber, and also other nutrients that are actually attached to that fiber compared with that isolated fiber supplement. Again, nothing necessarily wrong with that fiber supplement, but very, very different in terms of what you would get in fruits or vegetables. And so Yeah, that really is a big part of kind of the development of what we call NatureKnit commercially. And when we started the NatureKnit project and that research, we were really interested in helping fill that fiber gap. And so we started just by looking at where America's fiber is going, because nature makes lots of dietary fiber. Fruits, vegetables, nuts, seeds, grains. I mean, there's tons of fiber out there. And so if we're not getting enough of it in the United States, one of the very logical questions is, well, where is it actually going? Do we need to create more fibers in a laboratory, or, can we actually find this lost, missing fiber? And so that was really a fun part of the NatureKnit story, is doing our research on the American food system and understanding that actually nature makes lots of fiber, but during the food processing stages, really, especially for fruits, vegetables, and grains, we end up processing out a lot of that dietary fiber. And so juice is probably one of the primary examples. Grains would be another one. I'll talk about juice just because most of my research is on fruit, but juice is a really interesting area where juice manufacturers will press a fruit. They will retain most of the soluble components. Sometimes in the case of orange juice, maybe putting a little bit of that pulp back in, but for the most part, that fiber is completely separated. So as part of the NatureKnit research project, we started reaching out to food processors around the United States and eventually in Europe as well, started taking their byproducts and actually studying these in our own chemistry lab just outside of Chicago. And I think that probably is really the most significant part of the NatureKnit story, because we were looking for fiber. And I think what we found that was so fascinating is that when we would study these from a chemistry perspective and try to characterize these pummices, we found that they were rich in fiber, but they were also rich in diverse types of fiber. So again, going back to whole food fiber versus isolated fibers, there was numerous different fiber types that were there. So much more really perfectly reflective of the fiber we actually get as part of the diet. And then the other thing that we found is there would still be vitamins, there would still be minerals. And then very interesting to my PhD research, we found that there are polyphenols that are actually physically attached and bonded to that dietary fiber. So that's kind of when I guess, if innovators kind of have that aha moment, that was that kind of aha moment where we said, wow, these are really special fibers that we believe we actually shouldn't be throwing them away. And so let's do something great for environmental sustainability. Let's upcycle these. Great for the environment, but amazing nutritional benefits to humans as well. So that's really kind of a little bit more about the fiber gap, but also kind of the birth of NatureKnit was about delivering to people the type of fiber complexity, bound polyphenol complexity in a scalable way that really impacts both environmental health and human health as well.
It's such a great part of the story, the upcycling. We know consumers are increasingly aware, and it's something that they're interested in and appreciate. So we love that. Let's dive a little bit more into fiber-bound polyphenols. Can you go a little deeper into the benefits there?
Yeah, certainly. Yeah. So, and maybe just backtracking a little bit. So, kind of at the graduate level, most of my research was on fiber, fruit and vegetable consumption, and then about halfway through my PhD now. So, I think 3 more years to go. But now my research today focuses entirely on this kind of emergent issue of what is the relationship between dietary fiber and these unique polyphenols that are actually bonded to them in nature. So, kind of tracing back polyphenols, consumers are probably maybe increasing in their familiarity with polyphenols, really based on the popularity of the Mediterranean diet, which is kind of, people might say is kind of powered by polyphenols, and of course whole foods and fiber as well. But so polyphenols are known for really being the most abundant form of antioxidant found in the diet. These are very rich in fruits and vegetables, grains, nuts and seeds as well. And so fiber-bound polyphenols is kind of an emergent area within polyphenol science where these polyphenols in nature become associated with that dietary fiber. And so they're actually, the reason we didn't know about these until the last maybe 10 or so years is because chemistry tools had a very hard time detecting kind of where the fiber stopped and where the polyphenols started, because they literally look like they are bound and knitted together. It's actually where the name NatureKnit comes from, because if you look very carefully at fiber and polyphenols together, it literally looks like nature stitched them together. And so we're still exploring kind of what that means in terms of human health. But I think one of the really interesting things is that we know these polyphenols that are associated with dietary fiber have a unique benefit of being capable of resisting digestion, like fiber, in the stomach and in the small intestine, reaching the large intestine, which is also called the colon, and then really interacting with the gut microbiome. And so that is where we're most interested in future research and kind of unraveling the way that these fiber-bound polyphenols interact and benefit the gut microbiome. And so that's certainly a very key future area of research for us. But we do know that they do act synergistically. They're capable of kind of increasing the overall amount of fermentation activity from the microbiome. Polyphenols are capable of serving as kind of a localized antioxidant within the GI tract, which is also a very unique benefit to the colon. And then they're also able, through the activity of the microbiome, to be transformed into postbiotic metabolites. Not all of those are kind of fully understood yet. So when I say emergent, this is an area where there's probably decades of research to fully understand what's kind of happening between fiber and polyphenols. But it's really, really interesting because I've been fascinated by the dietary fiber gap, but now I'm also very curious if there's also a very unique polyphenol gap that actually is directly associated. When we're missing fiber, we're missing out on all of these polyphenols that are uniquely bonded to that fiber as well. So a very emergent area of research, and certainly several more years of research for me, but there are many other scientists in the field that are also very interested in this topic as well. So I would expect over the next decade, we'll have a much more robust understanding of the way that these unique fiber-bound polyphenols behave.
Yeah, I think I say a version of this every time we talk, but I just think you're in such a unique position because you're pursuing your PhD. I can't wait to talk in 4 years when we can say you just completed your PhD. But
me too.
Beneficial here is that your curiosity about the topic really comes through. Your passion about the topic really comes through, and it's interesting to see that, and then also how that translates into real science for the industry. So it's kind of the best of both worlds working together, and that makes it exciting, to talk about fiber and something you really paint that picture. But I'm wondering, from a research standpoint, we've talked a lot about your research so far. Can you put a fine point on what your hypothesis is about NatureKnit with regard to your research?
Yeah, absolutely. Yeah. So when we kind of finished a lot of kind of the paper research on NatureKnit, we did chemical characterizations of kind of numerous pummices and byproducts around the United States and also Europe to some degree. We built a formula that we felt not only kind of mimicked what someone would get in nature, but also really tried to kind of maximize fiber diversity and polyphenol diversity. We really felt, because there has been a link established between dietary diversity and microbial diversity and overall microbiome health, we felt like building a blend that was from real food, so upcycled from the real foods that people should be eating as part of a whole food diet, but then using chemistry tools to really try to maximize fiber diversity and polyphenol diversity. That really gave us kind of two things that we hypothesized and were interested in kind of scientifically validating. One was, we believed that based on that complexity, we could slow down the rate of fermentation. So fermentation in the colon in general is a very good thing. That's what produces all kind of the microbe magic in our gut microbiomes. But excess fermentation is known for being kind of a negative side effect. So consumers might complain with large amounts of fiber if they're not used to consuming that, or sugar alcohols or other things that can rapidly ferment. So we thought, compared to isolated and purified fibers, that complexity, long story short, would recruit more diverse microbes into that fermentation, and that would result in a long, slow, sustained, gentle fermentation profile. So that was one core hypothesis. The other one, which was kind of a very bold hypothesis, is that based on that complexity and the slow, sustained fermentation by the microbiome, we actually thought we would have a greater prebiotic effect than standardized isolated prebiotics. That is by far probably the boldest claim of those two hypotheses, just because really a lot of the isolated prebiotics are really designed to produce bacterial abundance and kind of produce bacterial diversity. So that was a bold claim, but it was something we thought, because by slowing down that fermentation profile, we thought we could have a prebiotic impact for such a longer period of time that we would actually produce much more benefit to the human GI tract and the microbiome compared with those isolated fibers. So those are really the two things that we hypothesized: a slower, more gentle fermentation profile, and then a more significant overall prebiotic effect compared to what the industry was using on isolated and purified fibers.
Excellent. And I love micro magic. I think we might see that show up in pages of Whole Foods magazine now, or get trademark that. Well, we've talked in a previous episode about some of the original research, the original study. And I know we have new research to talk about now, but before we do that, is there, any points you want to cover just to give us some context or an overview before we look at what's next?
Yeah, certainly. Yeah. Thank you for bringing that up. So we did publish an article in March of this year. It's in a peer-reviewed journal called Microorganisms. It focuses very much on microbiome-focused research. And so that study was on our original conventional version of NatureKnit. We now also offer a certified organic version. So that is really what our new research focuses on. Really, the actual product design of NatureKnit, the core hypotheses, the study model, all very, very similar. But whenever you change an ingredient, so when we switched from conventional ingredients over to certified organic ingredients, that changes things just enough. We also had some larger ingredient shifts. For example, the original version of NatureKnit uses quite a bit of conventional carrot as one of the primary fiber sources. The organic one uses organic kosher grape fiber. So when you make those changes, you really should do the science directly on that. And so we basically repeated that same experiment on the organic version. So in the original conventional study, we were very happy to see both of our hypotheses kind of validated through the study model that we used. And so basically we saw a very slow sustained fermentation profile compared to inulin and psyllium. It's really kind of probably the two most common purified isolated fibers that are used in food and nutrition today. And then from a prebiotic perspective, we saw a significant increase in overall bacterial abundance, and then also one of the key diversity measures, which is richness, how many unique bacterial species are involved with that fermentation. And then finally, one of the most important things you look for in a fiber study is whether it made short-chain fatty acids. Those are kind of the postbiotics that people talk about that have benefits in the GI tract, but also they reach the circulation and have benefits all over the human body. So those were the conventional results that we have and published earlier this year. And the organic study that we'll talk a little bit more about in just a minute was really designed to validate now just the new organic formulation. So kind of serving to kind of further validate the results from the first study with our new formulation, same hypotheses, and then just to make sure we were really doing apples to apples. When we did our comparison this time around, we did use organic inulin and organic psyllium just to make sure that we're giving all the fibers that we're testing kind of a fair shot. And so conventional in the first study, exclusively certified organic materials for the second study.
Excellent. So we've touched on the new study here a bit. Is there anything else you want to add to it, or do you want to just jump right to results and talk about the results of the study as well?
Yeah, we can definitely do that. I mean, maybe one thing I can do is just kind of remind the listeners, just in case they weren't with us the last time we talked about the conventional study, a little bit about kind of the way that we studied this. And so the study design, which is really the same for both studies, but the way that we do this is we use a validated SHIME model. So I think one of the questions people always kind of ask when they're not used to kind of microbiome studies is, well, wouldn't you just go right into a human study? And certainly we do have a desire to move towards randomized controlled trials long term, but these SHINE models are very unique in that they are validated to really mimic human conditions, but they allow studies into the active microbiome. And so in real human trials for the microbiome, because of course that person is living, they're alive, you can't dissect their microbiome as they're consuming your fiber. So you can really kind of see ingoing and outgoing, but in the middle you have what's called this black box effect. And so in order to overcome that, researchers around the world in the field of microbiology rely on SHIME models that are validated to truly represent human conditions, but they allow us to see in real time what is happening inside the active microbiome. And so if you're interested in the way that your fiber behaves with different bacterial species, it's a fascinating way to do studies and really get mechanistic insights into the relationship between your fiber and the human gut microbiome. So it's a validated model. We take fecal samples from healthy donors. The original study, we had 3 healthy donors. The organic study, we used 9 healthy donors. And so we have a screening process, and we collect fecal matter. We inoculate that into the system. And then that system is designed to perfectly replicate the human conditions from transit time to the right enzymes. And then we're taking the real microbiome from feces and putting that into the system. So then we can see in real time what happens when their microbes are exposed to various fibers. The other maybe unique thing is that we did a fiber matched study, which is fairly unique. So we are comparing the effect per 1 gram of fiber from these various sources. So it's a very fair way to study what is the effect of a gram of fiber from whole food diverse sources like NatureKnit. What is the effect of a gram of fiber from inulin and a gram of fiber from psyllium? So healthy donor studies, validated SHIME intestinal model, ex vivo design using real fecal donors to inoculate into the system, and then a fiber matched study. So we're giving all these fiber sources a fair chance to see what happens with the microbes that they like to interact with.
All right, excellent. So what did we learn?
Okay, so in terms of overall learnings, I mean, I think maybe one comment at a 30,000 foot level is we were also able to confirm both of those hypotheses again, which of course not always the case. So that's very exciting for a researcher to believe something, then to be able to validate that in the model. But really very similar to the conventional study, we worked very hard, although the formulas are different, to really again focus on scalability, but then maximizing diversity in fiber type and polyphenol type. So when doing that, although the formulas are slightly different, we really had very similar results. And so there are nuances, of course, there are differences in kind of certain microbes that at a 30,000 foot level, we reach significance on all those same primary measures. And so the things that we saw in the study that were very interesting, once again, we saw that slow, continuous pH drop. That really is kind of indicative of what the consumer would experience in terms of gentleness. So that was one of the most important things that we were looking at. So similar to the conventional study, the organic study, more donors this time, but we saw that continuous slow pH drop reaching significance against inulin and psyllium. And then in terms of short chain fatty acids, again, probably the most important result that you're looking for from fiber studies, we again reached significance in terms of total postbiotic short chain fatty acid production. Again, that is just an outstanding result for NatureKnit. Inulin especially is a very powerful producer of short chain fatty acids. And so to reach significance over inulin was a very, very powerful result. And again, the way that it does it is unique. And so those two results are kind of tied together. because the way that NatureKnit makes more short-chain fatty acids than a purified fiber like inulin is that inulin makes its short-chain fatty acids very rapidly because it ferments very rapidly. And so that prebiotic effect is over in just a period of just a few hours, where NatureKnit is fermenting for 48 hours. And so when we stop the experiment at 48 hours, NatureKnit is barely, but still actually fermenting. So it's kind of like the old Energizer Bunny commercials where it just keeps going and going and going. And so that's the reason we get these amazing results on short-chain fatty acids, because isolated fibers make short-chain fatty acids quickly, then they're done. NatureKnit is just slowly and consistently driving those short-chain fatty acid benefits over an entire 2-day period. When the experiment stops, it's still actually fermenting just a little bit. And then finally, when we get over to the prebiotic results, and so this is going a little bit deeper now into bacterial abundance, diversity, and richness. But we saw, again, like the conventional study, the organic NatureKnit formula was able to easily reach significance on overall bacterial abundance. So how the overall volume of healthy bacteria that was created through the fermentation activity, and then also in richness. Bacteria richness, I'll maybe pause on that just a second. It's an interesting look at bacterial diversity, but the reason it's important for NatureKnit is it's really a big part of our hypothesis, and really the reason we tried to methodically build so much diversity in fiber and polyphenol type into these formulations is because we wanted to recruit a much more significant part of the microbiome into that bacterial fermentation. So isolated and purified fibers, really, they like a smaller handful of microbes, and that is really what causes rapid fermentation. When something goes into your intestinal tract and maybe one major microbe like bifidobacterium or maybe 5 or 10, they like that, they can degrade it. That results in a rapid fermentation profile. What NatureKnit does, and the reason we built the formula the way we did, is because of its diversity, it recruits hundreds of different bacterial species, that healthy microbes that live in our intestinal tract. And so all of those different species come into the recruitment, and that slows down the fermentation profile and leads to a much greater long-term benefit. And so we see lots of microbes that were under the limit of detection become kind of at a much higher level after that 48-hour fermentation is done. So that was really probably, again, kind of a repeat of the conventional study. But whenever you change a formula, it's important to validate it with that specific formulation. So we were thrilled to really kind of validate those results. Again, there are certainly smaller kind of nuanced differences between the formulations, but for the most part, really validating all those same research measures again. Slightly larger study, 9 subjects this time. And so again, we saw consistency across the 9 donors, and that was also a very exciting part of the study, something we'll try to validate in future randomized controlled trials. But we do see with isolated purified fibers, you often have kind of a high responder or a non-responder, kind of mid-tier responders. Based on that diversity, we think that the human population is much more likely to have a consistent response based on that diversity. This is actually an area where Purdue University has been doing a fair amount of research too. And so we see that come to fruition in our studies, that consistent response across donors, likely based on that diversity that we built into the formulation. That was long-winded, sorry, but I'm happy to go into any more detail or certainly answer any questions.
Well, I think if we're going to have a long-winded answer, it might as well be about the results of a study. Just
True.
That's all good news. And let's talk about, in terms of, obviously, there are a lot of good industry-changing results that are coming through, but you also just got an award for the study research, correct? Can you talk a little bit about that? And feel free to run on about this one a bit too, because this is important, and I know it's hard to talk about yourself sometimes, but I would love to know. Tell me about the award, but I'd also love to know, what that means as a researcher.
Yeah.
To get that validation. How is that important also for the industry too, if you could?
Yeah. Well, thank you. That's a very kind question. This was recent. So I had the chance to, so the last couple of years I've attended the European Nutritional Genomics Conference. It's called NUGO Week. Doing my PhD at University of Aberdeen at the Rowett Institute. And so our organization is very, very involved in this European organization. So it's a really amazing affiliation across a lot of the nutrition universities across really across Europe. So this one was held just outside of Dublin, Ireland, and I had the chance to do just a poster presentation on our NatureKnit research. I was very surprised, to be honest, to actually receive the award for best poster research. It was really quite a humbling thing because this was one of those kind of academic presentations where, frankly, a lot of the researchers that you kind of admire, you read their research, you follow them. These are, this is University College Dublin, Trinity College, College Cork, Copenhagen. So a lot of the really amazing European universities leading microbiological research. So to get, most of them were judges on these panels, and so to receive an award from them was really, really very special. But I think what was very compelling to the European academics were kind of the conference theme was molecular approaches to sustainable diets. So I think what they liked so much were the scientific results were very, very compelling on kind of validating a lot of the things we know from epidemiological studies about the power of dietary fiber, but then also some novel things around fiber-bound polyphenols being present as well. So I think it was the combination of really outstanding scientific results, but also actually pointing back to nature, pointing back to real food, and then also having a huge layer of environmental sustainability. So again, not taking the approach of let's invent something new in the laboratory, but taking the approach of let's rescue something that is just being wasted that we think is good for the environment and we think is amazing for humans. Let's use science to maximize diversity in fiber and polyphenol type, and then let's use rigorous scientific methods to validate this. And so it really meant the world to me as a researcher. I think having your research also come to life in a commercial way is also very, very exciting. I'm very cognizant that not all researchers have that privilege, and so it's a wonderful thing to be doing scientific research, but then to be able to do something in our industry with that research and actually be able to offer brands and companies out there a chance to kind of take a part of this research and do something amazing again for human health and then also environmental health. And then finally for the industry, I also think I think our industry has continued to kind of raise the bar in terms of science, and that's something that I'm very passionate about. I think is very, very exciting. The NUGO Week conference is a true academic conference. There are very few industry people there, and so to have kind of an industry, publication there in the midst of a purely academic conference can be a little intimidating. And so to walk away also receiving a research award, for me, I think that's just a testament to the entire industry that we as an industry are really trying to raise the bar, doing much better in terms of maintaining rigorous scientific standards to test our products. So that was hopefully not only a win for me and for FutureCeuticals and Van Drunen Farms, really for the whole industry, that we are raising the bar and really trying to maintain that academic integrity through our research. So it was a very fun and certainly an exciting end to a week in Ireland.
We are just congratulations. We're so happy for you.
Oh, thank you.
Perspective for the whole industry, people, planet, good all around. So congratulations on that. I love it.
Thank you very much.
And we know you're not done, right? There's more to come, more years until the PhD, but also more work to be done, more things to be discovered. So what's on the horizon? Can you give us a peek into anything that you're planning or goals that you have for future research?
Yeah, certainly. Yeah. Thank you for that. I think it's interesting, whenever you have a research question and you do a study, you're always hopeful you can get that question answered. But then one of the things you find is that it also yields kind of 10 new questions that you would also like to answer. So kind of every study you end up saying, that was great. Now I've got 10 more studies that, if money were no object, I would now like to do. So there are some things that we are working on. I'll mention quickly on the academic side, at the same conference in Ireland, I presented an early part of my PhD, which is looking at kind of a systematic review of the health benefits of fruit and vegetable byproducts. So looking really beyond just nature and looking worldwide at all of the fruit and vegetable byproducts that are wasted and trying to help encourage our industry and others to think about valorization opportunities. So that is a piece of academic work that'll be published either later this year or early next year. And then in terms of NatureKnit, I mean, really, there's always so many more things that we want to do. So with NatureKnit, we are currently doing a bioinformatics study. And so one of the things, when you do these studies, the 16S RNA sequencing genetic data that we run, it produces just absolute mountains of data. So one of the questions is always, beyond bacterial abundance, diversity, richness, et cetera, how do we make this more translatable for brands? And so bioinformatics is a really interesting way to kind of take results like this and do large database comparison. So we're going to basically compare our results to existing microbe databases so we can get a bit more practical about what these results mean. Do they mean that we shifted the microbiome towards maybe a healthier state? There's a bit of scrutiny around, do we really understand kind of what a healthy microbiome is? But comparison to existing microbe databases say, what did, NatureKnit makes all of these changes, but what does it practically mean for a consumer that's taking NatureKnit? And so that study is really seeking to answer that question. That analysis will be done both on the conventional formula and the organic formula, trying to answer that question about what do these changes to the microbiome really mean in terms of practical human functional outcomes? And of course, that's also very predictive of what we might like to see in a human randomized controlled trial. And so that will also be used to kind of educate the way that we take this into future human trials. And then I think the other thing that's kind of come up, there's always emergent research over just the nuances of the microbiome. And so everyone's microbiome is a little bit different. So there are a lot of special populations that might benefit from kind of a unique formulation. And so children are a really good example where all of our current studies are done on healthy adults. We know that the child's microbiome that's really actively under development from maybe 6 months old to maybe 3, all the way to possibly 5 years old. So that's a population where their microbiome hasn't stabilized yet, but is actually still actively forming that stable adult microbiome. And so we're currently doing some research on whether NatureKnit could be useful for a population that doesn't yet have a stable microbiome, but is actually developing their microbiome. And in women's health, which has really emerged as a huge, very, very relevant topic for our industry as well. We're also very interested in some of the unique research on the female microbiome. And so on also researching on how NatureKnit could play a role in uniquely female health issues as well. So that's possibly another area of research for us. And then I've mentioned this a few times, but I think every researcher always hopes eventually that they'll get into more human randomized controlled trials. Microbiome research is a little bit unique in that the SHIME studies are really kind of the gold standard for mechanistic data of what's happening inside the microbiome. But when we get to more functional outcomes, I'm trying to say things like, what are these short-chain fatty acids doing in terms of improving overall human health? Or are consumers experiencing a more gentle fermentation that they actually feel inside their intestinal tract? Those answers will be sought through a future randomized controlled trial in humans. So that's probably the most maybe the most readily kind of next research steps we'll be taking over the next couple of years. There's always more to come. Every one of those studies will yield 5 more questions, and we will keep researching this until As long as we have a fiber gap and we're finding unique things that are great for humans, great for the world from a sustainability perspective, we'll keep researching these things as long as we possibly can.
Excellent. Well, you keep researching and we'll keep sharing the news. We love when you come and give us an update, and we can't wait, to hear the next one and find out what's new from the company. So thank you for joining us. Thank you for getting us up to speed, and we look forward to what's next.
Amazing. Thank you so much. Always a pleasure to be with you both. Thank you for your time.